Hello, in this video we will discuss about the epigenetics, which that means the above of genetics. It means the alteration of phenotype without the mutation in the DNA segment, while the addition or deletion of the molecule, for example methyl and acetyl. So it is known as the DNA methylation and acetylation. So this is known as the epigenetic we will discuss here. So here is a person which that's contain a different type of tissue. For example, the liver cell, pancreas and muscles as well as bones. Which that is their phenotype is different but their genes are same. So what that means? Why it's possible to produce the different type of organs? That means the switch off of the gene in the liver cell which that will produce the pancreas and express in the pancreas will suppress in the pancreas of liver gene. So here is the epigenetic mutation caused by the DNA methylation and acetylation to gene on or off without alter the DNA. So the environmental modification lead to DNA changes and express the new phenotypes. So in this way the methylation and acetylation within the life of the individual can be. So that means the different organs on the basis of the suppress and activation of the genes in the different region. If it will not work, for example, the liver which that contains that gene will activate, and if the activation of pancreatic gene will cause the cancer in liver. So that is important and easy to understand. So here is the chromosome in the nucleus of the cell, which that contain a histone protein 40% and 60% 60% protein and 40% DNA, and in this way it is nucleosome in the form of 8 subunit of histone protein with 4 types. So histone modification will be occur in the histone protein. So the methylation in the histone protein is very important, which remain unwanted gene expression will be inhibited through methylation in the histone modification, histone methylation. So is the DNA and the cancer causing, for example, gene which that can be hidden, hide or suppressed due to the methylation and DNA methylation. For example, gene 1, gene 2, etc. So the every cells in different tissue and organs contain different acetylation and methylation pattern. So every type of cancer in the organ is unique site of methylation. So that is important thing is that the methylation means the suppression of the promoter region. When suppress the promoter region will not promote the production of other growth factors. So that's why the uncontrolled cell division will be occur. So here is the epigenetic can be DNA methylation, histone modification through a methylation of the histone or acetylation. When acetylation will be occur, so that DNA will be expressed and non-coding RNA. For example, here is the transcription to produce the messenger RNA, and in this way here is the messenger RNA in the form of non-coding RNA, which that will be helpful for the gene translation. So that is important thing you should need to understand. So in this way here is the epigenetic and development we will discuss about. In this way here is the sperm and gamete for egg, which that is the gamete formation through meiosis to produce the egg and sperm, and after the fertilization both gamete, which that is the haploid, to produce the diploid zygote. So the every organs contain same genes but on or off of the genes by methylation and acetylation, and in this way the gene express and produce. So important things you should need to understand here: the zygote will develop the individual. This individual on the young age throughout the life will be changes the methylation and acetylation to lead the aging from the young to age due to the methylation of many genes. So on the other hand, important thing is that it can be reversible but not completely through some mechanism we will discuss end of this video. So here is the DNA. This is the DNA contain a C cytosine, guanine, cytosine sequence, which that is the cluster of sequence, cytosine and guanine. The cytosine and guanine here you can see this is the methylation is known as the CpG island. In this way the CpG island means the cytosine will attach with the methyl group is here. So this is the nucleotide, two nucleotides. This is a CpG methylation. So this is important things you should need to understand. This CpG island, for example, the CpG methylation will inhibit the some region, some things, and if the promoter region will be promoter here is you can see this is the methylation and it is the promoter region. This promoter region, for example, is the oncogene promoter region. If it will be methylated, so the promoter region will be not express the gene for the activation of the oncogene. So in this way the oncogene, the expression of gene will be genes repress off, but it can be expressed on the passage of time if the radiation or some things external stimulus can be or some age factor. So on the other hand here is you can see the 5-methyl cytosine. This here is the cytosine, the simply cytosine we will discuss. So here is the DNA methyltransferase A and DNA methyltransferase B, 3B, which that is use this enzyme and de novo methylation will be occur. De novo mean it is not present before, but it will be again the new methylation will be occur in the cytosine and the to become a 5-methyl cytosine. So here is the maintain the during mitosis, during the cell growth it will be maintained after the replication of the DNA. So after the DNA replication it will be maintained. It can be reversible, so it will the 5-hydroxymethyl cytosine through TET protein 1 to 4. It is the active, and the passive demethylation is also occur. So let's begin to understand important the mechanism for the methylation. So here is the you can see this is the cytosine. The cytosine with the DNA methyltransferase enzyme A and B, which that is formed to convert this type of the ring which that's contain basically is the carbon. So the methylation will be occur. DNA methylation will be formed. In this way, here is the methyl group will be attached, and again the TET protein, enzymatic protein, which will bind with the hydroxyl group with the methyl group. So the methyl group will bind with the hydroxyl group. TET 1, 2, 3, and these protein may promotes DNA methylation by binding to CpG rich region to prevent the unwanted DNA activity. DNMT activity. So the unwanted DNA methylation will be inhibited. While here the TET will again the different type of TET will be bound to form the 5-formyl cytosine. This 5-formyl cytosine to become a 5-carboxyl cytosine, and this TET is used again, and TDG and BER is used to produce the cytosine. This cytosine is looking like this, and here the TDG and BER, the base excision repair will be occur. Here is it can be directly the base excision repair is through these things. So the base repair can be occur due to the direct to form a cytosine. So here is the DNA methyl transferase 3B and 3A and methyl group attached to form a 5-methyl cytosine, and it de novo demethylation can be occur. So here is the zygote to pre-implant embryo and embryo formation, postnatal formation after the birth. So we will discuss here is the methylation pattern. So here is the global paternal demethylation in the zygote to move the postnatal after the birth. So here is the passive maternal demethylation will be occur, and this is the de novo demethylation, and after this the de novo methylation will be again after the blastocyst formation to become an embryo. So in this way, the de novo methylation, again the new methyl group will be added in the DNA for the suppress that gene which that is not required. And on the other hand, that was the before the previously required, but no in this time. So the demethylation in for CPG islands, which that is the basically methylation will be occur, and in this way again the maintenance and the demethylation in somatic cell will be occur, and the maintenance will be occur throughout the life. So here is the chromatin. This is the chromatin, which that is the part of the histone protein. 60% and 40% DNA, which that's contained here, is the acetylation and methylation. If the active chromatin mean the oncogene can be produced from the acetylation through oncogene activation, while the chromatin will be inhibited by the methylation to inactive or off tumor suppressor genes. Tumor suppressor gene will be off. So that is why this is the mechanism for the prevention of the oncogenes to progress the cancer. If the methylation to convert acetylation to progress, and oncogene will be activated. So here is the genetic modulation inside, and it is the innate, while the epigenetic modulation, which that is the adaptive modulation, and it can be through our digestive system, the microbiome in our gut, and the social interaction. If we will interact more, and we can confident more, and alternative uh, medicine, for example homeopathic medicine. On the other hand, the therapeutic drug, which that can lead to change the DNA methylation and acetylation to lead a disease others, while the alternative medicine is more better and improve lifestyle. And here is the psychological state, for example depression, anxiety, as well as the stress and fight and flight response throughout the life, which that is give the angriness. And here is the financial status. It can be a change our life, and drug abuse. On the other hand, the chemicals, for example smoking, cannabis, etc. And on the other hand, the other things which that is the toxin, the caffeine, nicotine, etc., and smoking. And here's the disease exposure through bacteria, as well as the virus and other protein factors. And this is the disease exposure to increase the epigenetics, and that means for the survivalship. So the epigenetic is the basic, is the genetic system which that is able to survive the person, and exercise and diet can change the epigenetic, and it can be negative or positive. So that is depend on our lifestyle. So that means the epigenetic, it is the above of genetics without the alteration of DNA. So thanks for watching. Please make sure to subscribe, like, and share, and subscribe.